additional guarantees which are good that our respective processing cells which are responsible for and ideally point nutrition and available. I find this our existing clinical set Dr. Kipit Respecting IPHC coordinator Madam Dr. Anamika Kapoor, she is unable to present contemporary by joining us in virtually. And our distinguished speaker is on the way actually. And our distinguished department, Dr. Nakuti Gordon, will come forward for our presentation and I will please our distinguished lady to manage the land virtually and enable to teach outcome. So, actually, because we didn't read that. 1 minute.
Nabudati, it's actually echoing. Can we do something about it?
It has to be in presentation mode. But when he's presenting now, yeah,
The audio is not very clear in my side too.
Yeah, and it's echoing, Nabudati.
Yeah.
Thanks to our hopefully, audio is echoing.
Yes, from my side, I mean, I can hear the echoes. It's not very clear.
Yeah, it's not very clear to me as well.
Now I am requesting our principal sir. Now it's okay. Now I don't think it will be before. Is it okay?
Yeah, better, I guess.
In this session, we have to discuss some unavailable resources, all the experiments, and I am this thing, but I didn't and I'm getting there by I am very good guys. So for my sorry, interested. Obviously, don't know this. I'm going to spend my weeks.
Hello, sound is not clear. Please check the sound system.
power system. সাউন্ডটা কমাতে হবে তবে কমাতে একটু সাউন্ডটা
somewhat good, but there is also some problem. Oh.
Ether? There was ether?
Now it has improved. Huh?
Now it's clear?
Yeah, now it's clear. Now it is very clear. Go ahead.
Scholars, students, and our colleagues, and others. I think it's our students and the colleges and the staff, the management of scholars, students, and students. I know this participants will get their so I some studies. And I mean, start also I'm saying I will learn more, especially I and others. I there are those nice reports.
I'm listing to our respective advocacy coordinator, Madam, to share some talks with us. Ready? Are you there?
Yes, I am there. Am I properly audible?
Yes, ma'am, yes, ma'am. Yeah.
Yes.
Okay. Good morning, everyone. I am at the very onset. I'm really sorry for not being able to be present physically on this very important international seminar organized by the Department of Chemistry on probing chemical insights into biomolecular advancement. And I'm very happy to see my former colleague, Dr. Shankar Prasad. They also in this seminar. Welcome, sir. Uh, I thank the thank and congratulate the chemistry department for organizing this in a very important time when medical research is not only important but being new avenues and new researches is coming up by the day. And also they have been very thoughtful enough to collaborate with the colleges with whom we have the memorandum of understanding. I'm sure that the discourses and the engagements that we today listen from the national as well as international speakers will be beneficial to not only our students but also the students coming from the other colleges, because seminars are so important to share the research with our peers. It is so important to for the students to understand how to present in seminars, how to work on their research and be inspired by new ideas. So I'm really hopeful. Really look forward to today's discourses and engagements. Uh, Thank you, all of you, and have a very good seminar today. Thank you. Do we have paper presenters? No.
So, please have to maintain at least one of the most critical challenges we must handle. Understanding and combating the application and the society. Even for the sun, standing at the investigation at the computer, and the sometimes he tries that sometimes I had now found I was just in a California. I have been following the feeling I was on the research and when the my current teacher is both and on time. I love underlying interview job is found in Suda that will lead from his and future public police capital. Data science contribution having data our application is technology and biological standards. We also found on the it has multiple other scientific research principles. are causing that not only significant factors, not only having programming even practices when the labor health come on not as even to start with safety, everything even very well. Hello, Yuri, are you there?
Hello?
Hello?
Hello? Yeah, I'm here.
You can start now.
Okay, thank you. So I hope everyone can see my screen and I'm audible. Hello?
Your screen is clearly audible to us. Now you can start.
Okay, thank you. So thank you everyone for, especially the organizer, for giving me this opportunity. And good morning, everyone. I'm Indrani. I work at Salk Institute. And today the topic that I'm going to discuss is evolution of HIV drug resistance. So before start, a brief educational background of mine. So আমি BSc করি শ্রীরামপুর কলেজ থেকে, then a master's from Scottish Church College. Then I did my PhD in computational chemistry from IIT Indore. After that, I moved to USA for my first postdoctoral experience. And then in 2020, I moved to Temple University, where I actually started working on HIV/AIDS. And in 2024, I joined Salk Institute as a staff scientist. A little bit fun fact about Salk Institute is named after Jonas Salk, যাকে ফাদার অফ পোলিও ভ্যাকসিন বলা হয়। যে এখন যে পোলিও ভ্যাকসিন বাচ্চাদের দেওয়া হয় অল ওভার দ্য ওয়ার্ল্ড, সেটা তিনি আবিষ্কার করেছিলেন ইন 1955, আই গেস, আজ থেকে 70 বছর আগে। তো তার নামে এই সল্ক ইনস্টিটিউটটা এস্টাবলিশ করা হয়েছে। ওকে, তো আজকে আমার প্রেজেন্টেশনটা দুটো পার্টে ডিভাইডেড। সো ফার্স্ট পার্ট ইজ হলো ইন্ট্রোডাকশন অফ দ্য ভাইরাস, যেখানে আমি হোয়াট ইজ দ্য ভাইরাস অ্যান্ড তাদের কম্পোজিশন কি, সেটা কি করে আমাদের বডিকে ইনফেক্টেড করে সেটা সম্বন্ধে বলবো। আর দ্য সেকেন্ড পার্ট ইজ ডিফারেন্ট স্কোপস অ্যান্ড ডাইরেকশন অফ কম্পিউটেশনাল কেমিস্ট্রি অর বায়োলজি ইন দ্য ফিল্ড অফ এইচআইভি, এবং সেটা দুটো অ্যাসপেক্ট নিয়ে আমি কথা বলবো যেটা পার্টিকুলারলি আমি কাজ করি। সো দ্য ফার্স্ট অ্যাসপেক্ট ইজ দ্য বায়োফিজিক্যাল অ্যাসপেক্ট, যেটা ইনক্লুডেড ইন মেশিন লার্নিং, এআই, and the second aspect is the chemical aspect, যেটা mainly included structural biology, mechanism, or mostly chemistry. So let's start with the virus and their compositions. So I guess because of coronavirus, everybody now know a little bit about what is virus or what that virus do. So virus is a submicroscopic infectious agent that replicates inside a living organism. So basically virus নিজে থেকে replicate হতে পারে না, তার জন্য একটা living organism like human or any other living organism এর দরকার পড়ে। So like SARS-CoV-2, HIV also একটা ভাইরাস যেটা আমাদের ইমিউন সিস্টেমকে অ্যাটাক করে মোস্টলি, এবং অ্যাট প্রেজেন্ট এইচআইভির কোনো পার্মানেন্ট ট্রিটমেন্ট নেই, এবং আমি বলবো পরে যে কেন সেটা নেই, এখনো কোনো এইচআইভির কেন পার্মানেন্ট ট্রিটমেন্ট আবিষ্কার করা যায়নি। সো কোভিড বা SARS-CoV-2 বা এইচআইভির মতো there are several kind of viruses present in this world. For example, hepatitis B, Ebola, influenza, rabies, rotavirus, herpes, etc. আর ভাইরাস যে সবসময় এরকম গোল রাউন্ড বলের মতো হবে তা নয়। ইউ ক্যান সি দ্যাট এখানে ডিফারেন্ট শেপস অ্যান্ড সাইজেস এ ভাইরাস আসে। নাও যখন আমরা বলি যে ভাইরাস ইজ এ সাব মাইক্রোস্কোপিক পার্টিকেল, মানে অবভিয়াসলি সেটা চোখে দেখা যায় না, বাট চোখে দেখা না গেলেও ভাইরাস কতটা ছোট, তো এখানে আমি একটা কম্পারিজন করেছি। তো ফর এক্সাম্পল, যদি আমরা একটা হিউম্যান হেয়ারকে মাইক্রোস্কোপের তলায় দেখি, তো সেটা সাইজ হচ্ছে অ্যারাউন্ড 550 to 180 মাইক্রন, যেটা এইরকম একটা স্ট্রিং এর মতো দেখতে হয়। আমরা যদি কোনো বালির দানা বা নুনের দানাকে মাইক্রোস্কোপে দেখি, সেগুলো সাইজ অ্যারাউন্ড 60 to 90 মাইক্রোমিটার হয়, এবং আমাদের যে হোয়াইট ব্লাড সেল বা যে রেড ব্লাড সেল আছে, সেগুলো সাইজ অ্যারাউন্ড এরকম হয়। সরি। কিন্তু এইচআইভি ভাইরাস বা যে কোনো ভাইরাসের সাইজ হয় এরকম, যেটা অলমোস্ট 0.1 to 0.4 মাইক্রোমিটার হয়। তো তুমি বুঝতেই পারছো যে এই যে এইচআইভি ভাইরাস বা যে কোনো ভাইরাস, সেগুলো আমাদের ব্লাড সেল বা আমাদের হোয়াইট ব্লাড সেল, যেটা আমাদের ইমিউন সিস্টেমের মেন পার্ট, তার থেকে অনেক অনেক ছোট হয়। তো এখন অবধি এই মানে গ্লোবালি এইচআইভি কেও অ্যাজ এ প্যান্ডামিক হিসাবে ধরা হয় লাইক কোভিড, এবং আজ অবধি টোটাল অ্যারাউন্ড 40 মিলিয়ন পিপল এইচআইভি তে মারা গেছে, যার মধ্যে অ্যানুয়াল ডেথ এখনো মোর দ্যান 1 মিলিয়ন। এবং যদি আমরা শুধুমাত্র ইন্ডিয়া বা সাউথ এশিয়া এই অ্যারাউন্ড এই এরিয়া ধরি, তো সেখানেও অলমোস্ট এখনো অবধি 6 মিলিয়ন পিপল এইচআইভি তে মারা গেছে, এবং ইয়ারলি এখনো 40 মিলিয়ন পিপল ইনফেক্টেড হয়, যার মধ্যে 1.5 মিলিয়ন নতুন কেসেস দেখা যায়। এবং শুধুমাত্র ইন্ডিয়া বা সাউথ ইস্ট এশিয়াতে এখনো 5 থেকে 6 লাখ লোক নতুন পার ইয়ারলি ইনফেক্টেড হয়ে থাকে। তো যে কোনো ভাইরাসের মতো এইচআইভি বেসিক্যালি দুটো পার্টে তৈরি। ফার্স্ট হচ্ছে প্রোটিন, যেটা হচ্ছে যে কোনো বায়োলজিক্যাল সিস্টেমের একটা বিল্ডিং ব্লক হিসাবে ধরা হয়। তো like viruses, we also have lots of protein in our system, and even any biological system has the lots of protein. Our second part হচ্ছে জিন, যেটা for HIV it's the RNA, but it can for different viruses can be either DNA or RNA. তো এই যে প্রোটিনগুলো, এই প্রোটিনগুলোর যে অ্যাটমিক লেভেল বিল্ডিং ব্লক, সেটা হচ্ছে অ্যামিনো অ্যাসিড, এবং ইউজুয়ালি 20 অ্যামিনো অ্যাসিডস থাকে। এই অ্যামিনো অ্যাসিডস গুলো কি হয়, যারা নিজেদের মধ্যে এরকম কম্বাইন এবং এই পেপটাইড বন্ডগুলো নিজেদের মধ্যে কম্বাইন হয়ে একটা বড় প্রোটিন বা যেকোনো প্রোটিন। এবার excuse me, can you please like mute your mic is on? Okay, so
somebody's mic is on.
yeah, okay, so এইচআইভির ক্ষেত্রে এইচআইভি কি করে আমাদের বডিকে ইনফেক্ট করে বা তার কিভাবে আমাদের বডিতে রেপ্লিকেট হয়, তার ডিফারেন্ট স্টেজেস। তো দ্য ফার্স্ট স্টেজ হলো এন্ট্রি, কি করে এইচআইভি আমাদের বডিতে এন্ট্রি করে। তো এইচআইভি ইউজুয়ালি ব্লাড সেলের আমাদের ব্লাড স্ট্রিমের থ্রু আমাদের নামে বোঝা যাচ্ছে যে এইচআইভি ইজ এ হিউম্যান ইমিউন ডেফিসিয়েন্সি ভাইরাস। তো এর কাজ হলো আমাদের ইমিউন সিস্টেমকে অ্যাটাক করা। আমরা যদি এইচআইভি কে মাঝখান থেকে কাটি, সরি হ্যাঁ, আমরা যদি এইচআইভি কে এরকম মাঝখান থেকে ক্রস সেকশন করি, তো সেক্ষেত্রে এইচআইভির ভেতরে একটা এরকম ক্যাপসুল টাইপের থাকে, যে ক্যাপসুলটার মধ্যে যদি এই ক্যাপসুলটাকে কাটা হয়, তো তার ভেতরে জিন বা আরএনএ গুলো এরকম মেসড আপ হয়ে থাকে। আর এই এইচআইভি বা এই যে ভাইরাসটা, এই ভাইরাসের বাইরে এই এনভেলাপ থাকে, যেই এবং এই এনভেলাপ প্রোটিনের থ্রু এই ভাইরাসটা হচ্ছে আমাদের হোয়াইট ব্লাড সেল বা আমাদের যেটা ইমিউন সিস্টেমের বিল্ডিং ব্লক, সেই হোয়াইট ব্লাড সেলকে অ্যাটাক করে। তো এবার কোয়েশ্চেন আসতে পারে যে তাহলে হোয়াইট ব্লাড সেল কেন এইচআইভি কে মেরে দেয় না যখন অ্যাটাক করে? বাট বিকজ এইচআইভি ইজ আ ভেরি স্মার্ট ভাইরাস। তো এইচআইভি কি করে, এই এনভেলাপ প্রোটিনের উপরে একটা সুগার কোটিং দেয়, যেই সুগারটা আমাদের বডি খুব নোন, এবং হোয়াইট ব্লাড সেল তখন ভাবে যে এই এইচআইভিটা আমাদের বডির একটা পার্ট, এবং তখন ও হোয়াইট ব্লাড সেল এইচআইভি কে ইন্টারঅ্যাক্ট করতে দেয়, বিকজ ও বুঝতে পারে না যে এটা একটা ফরেন পার্টিকেল, ও ভাবে এটা আমাদের বডির কোনো পার্ট বা বডি রিলেটেড কিছু। তো এইভাবে এইচআইভি তখন কি করে, এই এনভেলাপ প্রোটিনগুলোকে ইউজ করে ওর নিজের যে পুরো সেলটাকে আমাদের যে হোয়াইট ব্লাড সেল তার মধ্যে ধীরে ধীরে ইন্টিগ্রেট করতে শুরু করে।
Indrani, sorry for interruption. Actually, you may continue in English because there are some colleges with us. They can't understand Bengali properly. Okay.
Okay.
Okay. Thank you.
Okay. So yeah, so in that way, so maybe I can I can restate this slide again. So HIV integrates through this envelope protein, and the HIV first enters in our body with the bloodstream. And if you just cross section the HIV, then there is a capsule like this, and within that capsule there is a RNA mesh, or which is the gene. And when why the white blood cell or our immune system just instantly don't kill the HIV virus? This is because the envelope protein is coated with a sugar that is very known to our body. So the white blood cell doesn't understand that this is HIV is a foreign particle to our body. So that's why it allows the HIV to interact with the white blood cell. And with this envelope protein, it first sit on the surface of the white blood cell, and it started to interact with the surface of the white blood cell, and it start to insert the whole protein inside the white blood cell of our body. And so this is one of the first step of the HIV, that how it enters from our bloodstream to the immune system cell, or which is the white blood cell of our immune system, or start attacking the immune system. Now next is the reverse transcriptase. So what is this step means? This step means that when this HIV enters into the white blood cell, the outer coating is dissolved, and it's the microtubule that we has inside the white blood cell. This capsule use that microtubule as a road or as a highway to go to the nucleus of the cell. So when the microtubule is going through that, when that capsid is going through that microtubule, inside that coating or inside that capsule, the RNA that HIV has, that RNA try to become a fake DNA, and that fake DNA is occurred using a protein called reverse transcriptase. So why RNA need that fake DNA? Because our body, or mostly a human body, has DNA. So the aim of HIV is to integrate their gene with the host gene, and so that's why they try to make a fake DNA, so that this fake DNA can interact with the host gene. And when the capsule reaches to the nucleus, by that time, all the RNA, there are so many fake DNAs inside that capsule. So that part is called reverse transcription. So then the next step is integration. So what integration is? So when that capsule enters inside our nucleus, then that fake DNA that the HIV made, that integrates with our viral genome or viral DNA. And after that integration, our body cannot distinguish what is the foreign RNA, or what is the HIV RNA, or what is our DNA is. So when DNA replicates, it also replicates the HIV RNA as well. So this is how HIV replicates inside our body. So the next step is transcription. What transcription means is that after the viral DNA integrates with our DNA, then it replicates, our body naturally replicates all the DNA. And with that, the viral DNA also replicates. And after that, there is a viral protein that from that replicated DNA, it make more RNA that is needed for the virus to have a more virus particle. So using this viral protein from the replicated DNA, there is a lots and lots of viral RNA was produced. Now this process is called transcription. Now the next part is translation and assembly. So again, using the same viral protein, when there are many viral RNA are produced, they kind of assemble and try to be deposited in the surface of the white blood cell. And the nucleocapsid, which is another protein that try to make like a coating around that assemble of RNA. So that whole system is called the immature viral particle. So this particle is still not infectious, and when there is enough RNA that was present in that capsule, the nucleocapsid did is they just block the whole process, and that immature particle started to be out from the white blood cell. This process is called translation and also the assembly of the immature viral particle. So then the last step is budding and maturation. So when all these immature particles go from the white blood cell, there is a protein called protease that kind of use like a scissor, molecular scissor. So what it does, it cuts different amino acid inside the immature particle and started making a coating around the RNA. That coating which initially enters into or invade our nucleus or the cell. So when that protease finishes the coating around the RNA, then the virus become mature and become infectious. This is how like a whole this is like a HIV cycle inside a human body, when how HIV enters and replicates and infect our whole immune system. Now the question is, like, as I have mentioned, that HIV doesn't have a proper treatment yet. So why is that? This is because, like, with COVID, we already know that COVID has different strains, right? Like virus have different strains. So these strains comes from the mutation. So what any virus do is they try to survive, and they try to change ourselves so that they can survive. And that change is called mutation. So what mutation exactly mean is that, as I have mentioned, there are different proteins, and the proteins, the building block of the proteins are the amino acids. So if there is an amino acid in a protein chain that change to a different amino acid, that is called mutation. And so basically how the HIV virus change itself and fight against the treatment. So it makes some specific mutation that kill the treatment or kill the drugs or medicine that given to a patient, or those mutations are called the drug resistance mutation. And these drug resistance mutation are mostly the topic of my interest, or that I mostly work on. So now I will talk about what my work is, specifically the two aspects that I usually work. One is the biophysical aspect or the machine learning process. So basically, as I have mentioned, that there are different drug resistance mutation, and those are found in different patients all over the world. But like after starting the treatment of a patient, when this drug resistance mutation usually appear, what is the timeline of it? So usually, like we find out there is no proper like study. There are many papers that reported different timeline of different drug resistance mutation, but there is no like a proper class or category of it, or there is no way to predict that how or when this drug resistant mutation will appear. So we have a team, or I have a like a colleague who modeled a machine learning tool that can, so that machine learning model is trained over all the patient sequences. So all the patients currently in the world who have HIV or go through some treatment, their blood samples, their sequences are usually most of the time deposited on some databases, and we have the access of some of those databases, and as well as the access of some personal patients throughout different hospitals of USA. And from those databases, we trained our machine learning model so that they can learn the behavior of this drug resistance mutation. And using those training method, we try to calculate the timeline of different drug resistance mutation, and we find that our model can accurately predict the timeline of different drug resistance mutation in HIV, and the accuracy is from 75 to 90, or almost greater than 90% in some cases. So how that will help? So the first that will help is, so when a clinician or a doctor treat a patient, looking at different drug resistance mutation is a very time consuming and very costly. It takes years. Sometimes you don't get volunteer of patient, or sometimes the patient don't complete their treatments. So with this method, we can have easily interpret, or at least give some assumption to the doctors or the clinician, that if they started a particular treatment, when and how these mutations will appear. At least give a more or less assessment, so to different patients, depend on various kind of drugs. Now the second aspect is the chemical aspect, which is mainly based on the structural biology or the mechanism of drug resistance. So as I have mentioned before, that the integration process, which is where the viral DNA is integrated with the genome DNA or the host DNA with our body. So this integration process is coordinated by this protein called integrase. Now, this integrase protein, so to block the work of this integrase protein, there is a medicine, or there is a class of drugs which called INSTIs, or integrase trans transfer inhibitor. First, these INSTIs are invented in 2007, and from that, like from 2012, 13, 14 up to 2021, there are several medicine or drug is a drug is going through trials and get FDA approval. And the latest FDA approved drug is cabotegravir, which is approved in 2021, or we call it in a short CAB. So this CAB drug is currently from 2021. It is given to HIV patients all over the world. Now we see that from 2021 to 2025, which is just 4 years, so we don't have a lot of data. But what we can find, that because of those patients who are taking this CAB drug or CAB medicine, they usually found this, particularly 3 mutations in their system. These are position 138, 140, and 148R. So these 3 are the most focused mutation that we are currently working to understand how this mutation leads to the resistance of this new drug, cabotegravir or CAB. So let's look at the structure of the integrase. So one thing that our team is mainly do, or mainly the main feature of our group, is to look to model or to to image the HIV or human integrase structures. My mentor Dimitri Limkis is the first one who first make a cryo-EM image of the whole HIV integrase. So it's a very big hexadecamer structure. So this is one part of the HIV integrase structure, and in this part, if we zoom in, where this is, we call the active site, when there is 2 magnesium, and when a drug or a medicine is given to a patient, that medicine come and bind to this integrase, and that's how that it blocks the integration process of the HIV. But what happens if there is a drug resistance mutation at the active site? For example, this Q148R, or which is the mutation to 148 arginine, that started interacting with the active site, and that block the drug benefit or the work of that CAB needs to do using this drug resistance mutation. Now the question is, what is the mechanism of this drug resistance mutation, how it is affecting the drug binding or affecting the whole drug resistance. So what my work is mainly looking at the mechanism of this drug resistance by using molecular dynamic simulations. So molecular dynamic simulation is a quite old method to look at the dynamics of a system. It can be applicable to the biological system, materials, or any kind of bigger systems. So what molecular dynamic is actually, so basically when we have a structure like a cryo-EM structure or X-ray structure, that is a one snapshot of a protein. But within our body, usually all these proteins are dynamic in nature. So molecular dynamic simulations help to capture the dynamics of those systems. For example, like now how we simulate those biological systems. So what we usually do, we took the protein. So let's say these colorful ribbons inside this box is the protein. We took the protein, we put it in a water box, because inside our body or inside any biological system, they are mostly submerged in the water, right? So we put the protein within a large water box. And our bodies also have ions as well. So this pink and this green ball, these are the ions that mimic the whole biological system inside us. So now when we make the initial input, we simulate it through various nanoseconds to even microseconds. What simulation means, like you can imagine it as like a game simulation. Like I know many people can play games, right? Video games. So where in video games there are humans and everything is simulated. So this is something kind of like that. So yeah, so we simulate this whole system for a nanosecond to a microsecond level to understand the dynamics of the situation. Like within that simulation, so this is, I'm focusing only on the active side, and you can see that active sites are like jiggling all the time. So this is how the dynamics of a system, or usually inside our body, the proteins are jiggled. They didn't like static inside our body. So we try to capture the actual biological process through this simulation. So so using these methodologies, we try to understand the mechanism of this resistance of carb. So we find that this mechanism or this resistance happen in 4 steps. So what the step one is? Step one is that this Q148R mutation, it started to interact or started to drag the magnesium of the active site towards it, because it has a delta positive and positive negative charge interaction. Usually if the positive negative charge interaction, they both attract each other. Now, when this magnesium is attracted, the magnesium and the carb coordination is destroyed in the second step. Due to the destruction of this coordination, the whole carb is shifted from its original position. That is in step 3. And due to this shift, the other coordination between magnesium and oxygen are also destroyed. So this is how a drug resistance mutation at the active site can diminishes the benefit of a drug or a medicine that is given to a patient. And that is how HIV like wins over the treatment. And that's why it's very difficult to create a proper treatment for HIV. And there is another thing that so every virus did go through the mutation. Then why HIV is so special? Because the rate HIV mutate, that is very, very high, like compared to like a different blood cell or different host, it mutates million times more faster than its host, or even from a different virus, it is 100 to 1,000 times faster. So that makes HIV very difficult to make a permanent treatment, because whenever there is a treatment, after a few years, you see there is a mutation that kills the treatment. So that's the major problem currently. Till like after like HIV, I think first detected in 1982. This is almost like more than 40 years, but still we couldn't able to find the proper cure or permanent cure of HIV. So what we learned today? So we learned that virus is a subatomic infectious agent that replicates inside a living organism. Like other viruses, HIV also consists of protein and gene, which is RNA. HIV changes itself to fight against different treatment, that is called mutation, and specifically the drug resistance mutation. This drug resistance mutation can have different timelines and within a patient, depend on different treatment techniques. And using a machine learning model, we can able to predict a more or less accurate timeline of different drug resistance mutation, and the accuracy level can be from 75% to 90%. And using a molecular dynamic simulation, we can hypothesize or predict the mechanism of drug resistance that may help to design a more accurate or proper treatment in future. So last but not the least, I like to thank my mentor, Dimitri, Tao, and Avik, who are the postdoctoral candidate in my group, and we closely work with them, the rest of the group, and all our amazing collaborators, because these are multi-collaborative projects, and so without them, these projects cannot be possible. The funding agencies, and particularly the California HIV Research Program, who personally fund my work. And thank you all the organizers for giving me this opportunity to present my work. And I'm happy to take any questions if you have. Thank you. Hello.
No, yeah. No, and so I'm going to. Okay.
The audio is not very clear to me. So if anyone can, if anyone have any question, they can. I mean, it will be helpful if it writes on the chat. It's not very audible to me.
Okay, Nandini, I'll try to write it down. So if anyone have any questions, please write it down in your chat box. I'm asking it for the Ranchi University College students who are online. Please write it down, and Navadip College, please. And I am going to take the questions and I'll write it down for you. Is it
Thank
okay?
you. Yeah, thank you very much. I mean, I can hear you like not very clear, but I can hear you. If someone can ask me a question or someone in the online, I can happy to answer.
So Inani, I am going to question you in this format only. I'm not writing my question. First of all, it's very nice presentation. Thank you for your vivid presentation. And just I have only one question. You showed that gap, oxygen and interaction, right? So that oxygen is from gap or from our bloodstream or from the white blood cell.
Sorry, oxygen?
Yeah. So the interaction between Mg and oxygen, right?
Oh,
So,
okay. So this carb structure, when it is delivered to the patient, it is coated. So these carbs molecules are not so like I showed this carb structure. It is coated with some non-interactive agent so that it doesn't interact with the bloodstream or any other thing. So when it close to the integrase or the integrase protein, then only it uncoat and then the carb releases or the main drug releases so that it doesn't directly interact with the other part of the body. So those are designed in such a way. Any drugs is mainly designed in such a way so that they are targeted to a particular protein. It only releases the drug when it is reached to the target.
Okay. And so Mg was in which coordination at the first time, and then you told that weakening of oxygen and Mg bond. So it is in 6 coordinated system or any other coordinated system?
That's right. So magnesium is usually 6 coordinated, but then when the drug resistance mutation started to interact, it started to lose the coordination one by one.
Then it gets stabilized by three coordination, four coordination.
No, then the water molecule in our body takes that coordination.
So it takes like the energy.
Yeah, that is easier because there is a lot of water molecule that is easier to substitute.
Yeah, I understand. Yeah. So thank you. We don't get any questions. Yeah. Hello.
Yeah, thank you. Thank you very much.
Okay. Thank you.
Thank
So thank you. Yes. Okay. Thank you then, Nani. You can clear this out. Yeah.
Thank you.
Now, someone is so from Rachi Municipal, someone wants to ask a question. Please write down your question in message box. We'll send it to Indrani and get the
Sure,
answer.
sure. Write it down. I will answer that in the chat box.
Yes. Thank you. So we'll proceed forward for the other section. In 10 minutes, we are joining again. After a tea break, we are joining again after 10 minutes. Hello. এই এক সমস্যা আমাদের এখানে। আমি তো লিখলাম না। Hello. Please, please silent, please. আমি কিন্তু এটা আমার টেবিলে করেছি। হ্যাঁ, আমি করব, আমি কমিয়ে করে রাখছি। তাহলে আমি তো আমার মোটিভেশন করতে পারবো। কিন্তু পরে এটা তো একদম নেই এটা না। ইন্টারনেট লিংক কি করে জয়েন করবে এটা তো? হ্যালো, am I audible? Hello? 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 Pragati, Lakshmi, am I audible? Hello? 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 I কোথা থেকে জয়েন হয়েছে? Am I audible? Tushar, am I audible? Okay. Then we should start. And he brings forward with the department, Dr. Navagun Gordon, to share some few words in terms of I did not and also aspect. And sort of should say 2 words. Department of Chemistry to see both offline and online mode. And we all I will wait, just concepts first. Otherwise, range, I'm going to guide. Yes, you are doing it. Because I think really bored about these standard content and black and all the scientific names and also say one thing about understanding that I don't really right now. But and see that knowledge in chemistry, methods of thinking, there's some data, which are data memories, always favorite, and how to say that? Like this is character of my knowledge, which is very good. One of the very officers started at class. How oxygen vaccine and how it transfers to our cells and structures on the biological processes and by the quality choice and begin by this is not fun. So on the things I was and that is why I also was excited about all the wonderful chemistry and therefore biological science to understanding of chemistry. Then I started a new program for this business chemistry and biology. To explain substance somewhere on the and we have added to this program. Our little dreams from a small community of we were starting to on the one membrane of the medicine and but all the investment and to start English of them. And probably statistically, in one of the studying regeneration of organisms suffered they were necessarily in environmental exposed that when the amendment and the fourth bases are the minor same throughout the region. Thank you. Nobody. We have Commissioner Dr. and we will start our private in the Department of Biochemistry and Biology in the Institute of Medicine. We often use space shallography and molecular biology from the Bhavan Institute of Medical Physics. We extensively experience and research Dr. Bell specializing in the study of human development, one biology, and nutrition. Multiple government mandates like NCRT community international collaboration with the Endocrine collaboration initiative. More than 45 peer-reviewed publications and 5 recent breakthrough work in the carbonized biology marks a significant national contribution in linking the journal. Dr. Bose is a recipient of peer-review fellowship and Young Scientist Award and has mentored scholars while actively collaborating with global scientific programs in oncology and environmental biology. Expertise on the molecular characterization of human emergency of cancer. Science in India is the most difficult consider in the molecular oncology and is to understand the disease fundamental chemistry. These are therapeutic innovation.
So, yes, everybody. I'm listening to chairs. So, good. I need how I do. And then I will then we are not. I so the sun doesn't balance. If you mean biology, it's not because after all, one molecule, one point, interacts with the other, so that's the other structures based on the structure of the molecule, which follows some principles of physics. And we know that we say that like everybody gets some sound simultaneously, and they have to match up each other. So understand the infinity of the violence is sometimes need the help of the mathematical model. So I got some sort of model. Even if we have no details, like we have one map, one model. And we are trying to find out which one we do not do the God. So we are the last so small values and value are random molecule and theoretical study. We find the things that's a thing is we like that. Because now we are in the era where everything is doing something in technology. Basically, there is a video taking the this is the complement each other. And this complement is the complement biology by this complement like that. And it's in the very yeah. If you don't
ऑनलाइन ज्यादा क्यों नहीं हो रहा?
When the user while we are talking about the back, while we are using the back, this is a great important one. I think if we actually run very nicely, and this one structure of the block, and because learning often we have one molecule developed to take long action the fibers and now also develop and you take one by one and try to find the time. Zero target to so serious take finding them. So after this implementation molecule again which one so the pattern needs a mixture of nature, the chemicals and immunosensitizer and ability. It's like the other, like Another problem to study from here has demonstrated minor. This kind of work needs from activity. They say that we can check how much movement is required. Let's say these are the Western block. Now, the important brand of this what is the these particular companies are intention the they are released and beginning to that way the position that's the thing. There are different models. Particles are higher and I have the connection. We have So, Rajendra, really good afternoon. For the beginning, carbon and the only one which is right now you can engage unable particles like I can actually charge particles and those particles are moving from that interface and again we have taken the necessary again we check the similar out of the account is divided by then our I'm not yeah, I don't the same way of doing research and reporting that we are doing. So that we are going down or going up. That team that answers to the so that they are figured yeah, right is article that is speaking of the particle therapy see and then gradually it's down. If you only get one, and cancer we can move to the of the chart but I don't know. The problem is that thank you. When it needs to take inside the and after calculating the size of that, the community of this capital accordingly done so that we can here you see the means actually that's why this particle can I think you know regularly number of cases we take with the particle connectivity. Which is going into the first particle therapy practice the although particle therapy are targeted, how do I say that it's done? It will be one of the change it up. When you have long term basically how do you explain the difference? Now it is Private sector and so yeah, I will say that this migrate from there and some again young and communicate and and say that you migrate from one these are the again these are the total and first say you migrate from one place to another. So if you have an entity to higher gradually we have I can but activity is very important. Actually, the health of if you target those then that will be the so these are the ways which is the right nothing means the activity. Reduction of activity means I'm not going to if you combine the identity with this particular this particular so come to the
Please write the chat box. And we are sorry for the power surge and Now we have a speaker, Dr. Shamini Bharat. Shamini Bharat here. Shamini, are you there?
Yes, I can hear you.
You can present now, please.
Okay, I'll be.
Just share the slide and just put it in a proper mode and then we will start.
Okay, can you see my screen?
Yeah, we can see your screen.
Am I audible?
Yes, I will. Okay, start with it.
I can't hear you properly.
You can hear us properly or you don't hear us properly.
No, I don't hear you properly.
Okay, that's okay.
Should I start now or?
It's not audible. Now it's audible.
Yes, a little better.
And I'm not going to That's it, explains that developing and releasing and violence using underlying active frequency. This results in disease to the of their conversation, seeking the line on how the nature on using the use of action to undermine even the public that is an extremely negative way balance education. We think about publication and we will check them. Further continuation development advance and analyzed in the system. In this you can start now, Samir.
Okay, if I don't hear you properly, so I'll be starting. If I am not audible, just tell me.
Okay.
Okay. Thank you very much for inviting me to deliver this presentation. So today I will be talking about revealing molecular mechanism of photosynthesis by using advanced spectroscopy technique. So by considering the audience today, so I will try to be as simple as possible, but if something is not understandable, just ask me. So in next half an hour, I will try to give you a glimpse of this spectroscopy technique and how we utilize it to understand the molecular mechanism of photosynthesis. Now I will start with photosynthesis, which we all studied in school. So this is very basic biology, where we know that some organisms like cyanobacteria, algae, or plant, which is more familiar to us, they do photosynthesis. So in the presence of sunlight and carbon dioxide and water, they harvest chemical energy, and as a byproduct, oxygen is evolved. Now this process, all we know how it happens in leaf, and if we see inside the leaf, there is this thylakoid membranes, and these thylakoid membranes are embedded with these photosystems like this on the left. So let me get the pointer. Yes. So on the right, you see this thylakoid membrane and these embedded proteins, which are super complexes like photosystem 2, cytochrome B6F, photosystem 1, and so on. And we all studied these processes, how it happens. Now, the point is here, so under sunlight, these photosystems absorbs light, and then a charge separation happens at the reaction center, followed by these electrochemical processes leading up to the formation of NADPH and ATP and all. Now here in spectroscopy, where we are interested is what is happening inside these photosystems. Now if we look into the photosystem to super complex structures, this is the structure we have. So we have many proteins around this. So proteins are basically made of amino acids. So these are polypeptides, as you know. And these polypeptides from in these proteins, they have these 3D structures. Now these individual proteins, so there are many around this reaction center. So reaction center is where the charge separation happens. And around this reaction center, we have all these light harvesting complexes. Now, at the periphery, we have light harvesting complex 2. We have 3 here and 3 on the back side. And we have other complexes here in between this peripheral and the core complex, which has like different names, CP47, CP43, and in between we have CP29 and all this. So basically what these proteins do. So proteins are amino acid, as I said. So they absorb in the UV. Now we know sunlight is visible. So to harvest this visible light, we need some pigments which will absorb in the visible. Now, as we all know, why plant leaves are green? Because of chlorophyll. So chlorophyll is the pigment and also carotenoids. They absorb the visible light from sunlight, and then they transfer the energy to the next chlorophyll, from next chlorophyll to the next protein, and like that it went to the reaction center. Now, if you consider a football match, the ball starts at the middle and one player passes to the another one, and at the end it reaches the goal post. So the goal post here is the reaction center. So, and this photon first absorbed by the chlorophyll, transferring through many chlorophylls in between and many proteins, reaches the reaction center. And then charge separation happens, followed by different reactions. Now, in ultrafast spectroscopy, we are interested in this photon absorption by the chlorophyll and how it transfers the energy to the next one, followed by when the photon reaches the reaction center. So this is all we know from the textbooks. So these two proteins, CP26 and CP29, that I will come back after a few slides, because these are very important. This bridges the peripheral proteins to the reaction center. Now you may ask that this is all we know. So what is new here you are trying to study? Now let's consider a normal day. So we have normal sunlight, and then this PS photosystems absorbs the light, followed by the electrochemistry to do the photochemical quenching. So photon, then it quenched, and then it is harvested at chemical energy. Now, if there is high light, then there are many chlorophyll around this reaction center. So everyone is absorbing. Some of them will transfer to the reaction center, and the charge separation happens. Now the reaction center is busy at that time, but there are many other chlorophylls which went to the excited state from the ground state. So now it cannot transfer to the reaction center. If it cannot transfer to the reaction center, then the chlorophyll remains in the excited state. What it will do, it will donate that electron to some dissolved oxygens to form reactive oxygen species. So these reactive oxygen species are dangerous for these organisms because it will destroy these photosystems. So what these plants do, they dissipate this excess energy through different forms so that it does not generate the reactive oxygen species, so that it can protect itself. So therefore, these photons is not utilized for chemical energy conversion, but rather it is used for non-photochemical quenching. So this is the term we will be using frequently in the next few slides. So this is the NPQ, which also called photoprotection. Now, why photoprotection? Because it helps the plant from not generating reactive oxygen species so that it can be healthy. Now, during day, if there is a cloud, let's say, so sometimes sun is there. When it comes under cloud, so there is less sun. So it switches between photochemistry and photoprotection depending on the sunlight. Now, if you know the exact mechanism of this switch, then you can harvest more light. So there are recent studies where people are saying that if we tune this photoprotection mechanism, then you can harvest this light in a better way so that the crop production can be increased. So in the tobacco plants, they saw that 15% greater plant production can be achieved by tuning this xanthophyll cycle, which I will come later. However, these photoprotection mechanisms are not very clear to us yet, like how this chlorophyll and carotenoid interact between them and dissipate this excess energy and then switch back to the photochemistry part where it can harvest. So light harvesting to quenching. This process, it is not very clear to us yet. To study this process, we will be using spectroscopy. Now, to use spectroscopy, let's go back to our very basic BSc spectroscopy course where we all studied Jablonski diagram. So we take a standard Jablonski diagram where we have a ground state, which is S0, and excited states like S1 and S2. Now, when there is light, we all know it absorbs the light and instantly it goes to the excited state. Now, how instantly it is? Now, in the ultrafast spectroscopy domain, the time scale that we are talking about is femtosecond. So it absorbs the light in femtoseconds. And what are the processes happens in the excited state? We all are familiar, like internal vibrational relaxation or internal conversion. And from excited state to the ground state If it emits photon, then we call it fluorescence, and the time scale is in the order of nanosecond. Or it can go from singlet to triplet through inter-system crossing, that is ISC we call. This is spin forbidden, so this is a slow process, and from there, if it goes to the ground state, we call it phosphorescence. Now, these are all the processes that can happen in a molecule, and the time scales, as you can see, in the order of picosecond, nanosecond. So we need a time resolved spectroscopy in the order of femtosecond, picosecond, or nanosecond to study these processes. Now, what are the molecules? I said chlorophyll. So here is the chlorophyll structure. So this is a porphyrin ring. You have a magnesium inside. And if you look at this electronic structure, so it has a S0, and from there it goes to Q or B band. So depending on that, you get the absorption spectra here. On the x-axis, this is wavelength, and the energy gap, all we know, equals to hc by lambda. So as lambda increases, energy gap decreases. So this energy gap is less, so the lambda is higher. So absorbs around 650 or 700. Whereas the blue one is higher energy gap, so absorbs around 400 nanometers. So this is the two bands we have in chlorophyll. Now, if we excite this chlorophyll, so we pump a light, let's say laser, in this region, then it goes to the excited state, and when it comes back to the ground state, it emits. So we measure the photons, what it emits, and we plot it against the wavelength, and what we find is the chlorophyll emits around 680, 690 nanometers. So this is the emission we have. Now, how to measure this emission in time domain? So time resolved spectroscopy here. To do time resolved fluorescence, we have a laser here. Don't go into the detail, it seems complicated, but I will try to make it more simple. So we have a laser, you excite the sample, and you collect the photons that it emits through the streak camera. And there it is basically, you have this phone camera, you have many frame rates, so that you can make the video also. Here, we are talking about in picosecond resolutions. So with this camera, we get a 2D map. So on the Y scale, we have a time which is in picosecond, and on the X scale, we have wavelength in nanometer. Now in this 2D map, we can get two informations together. One is the spectrum, like the energy of the photon, so in which wavelength it emits. And the second information you get is what is the dynamics. That means how long that electron stays in the excited state. Now it decays slowly, you see, in 2000 picoseconds. That means 2 nanoseconds, it is decaying most of like 80-90%. So by using this technique, we can study that which molecule, how long it stays in the excited state. Now we have another molecule, that is carotenoid. So this is the carotenoid here, like all we know carot. So this is beta carotene. So simple, these are the carotenoids we have in nature. Now in this carotenoid, if you look at the absorption spectra, it absorbs in this 450-500 region, because from S0 to S2, and this is the absorption spectra. However, this carotenoid does not emit because it has very low quantum yield, and in lighted Western complexes, we do not observe emission from carotenoids. So in that case, we cannot use time resolved fluorescence or photoluminescence to study carotenoid. Now to study these carotenoids, we use pump probe spectroscopy or transient absorption. So what it does, basically, it measures the change in absorption by using two laser sources or two different lights. One is pump. So you illuminate the sample with the pump, it goes to the excited state. So from S0 to S1, let's say. And then we send another light, which is probe beam, to see what are the processes happening there from this S1 state. Now, in general, there are three processes that can happen. In the presence of probe, we are monitoring the ground state bleach. So we see the change in the absorption, that is delta A less than 0, and stimulated emission if it emits, or excited state absorption. So in the presence of probe, it absorbs from at the S1 level and goes to the higher excited level. So among these three processes, what are the processes we see in carotenoid and also in chlorophyll, and between them how the photophysics is happening? From there we can know what are the molecular mechanisms that is happening in photosystems. Now let's come back to the photosynthesis. So now we know we can use time resolved fluorescence, we can use transient absorption to study chlorophyll and carotenoids inside the photosystems. Now, in the presence of highlight, I said it goes to the photoprotection or non-photochemical quenching. So what are the molecular parameters that is influencing the non-photochemical quenching? So there are three parameters. The first one is drop in pH. Now, we all know in lumen, the proton accumulates, so the pH drops. So what happens when pH drops? So when we see this is LHCSR, so this is one of the light harvesting complex. So we took this complex and changed the pH externally to see how it changes upon sensing the pH. So the black one is the protein at pH 7.5. What we can say is normal physiological pH. And the red one is when we change the pH to acidic. So that is pH 5. And on the x-axis, we have the time scale in picosecond. Now you can see from the black, this is the lifetime. So at 2 nanosecond, it is still in the excited state. Whereas in case of the red, you see this is decaying faster. So what it means is, in the presence of protons, like in lower pH, the chlorophyll quenches faster compared to when it is at pH 7.5. Now we know it is quenching faster, and these are isolated proteins, so it is not transferring to the reaction center. So chlorophyll is quenching, that means it is transferring to somewhere else. Now we need to find out where this chlorophyll is transferring to instead of the reaction center. So that is how the photoprotection is happening. Now there are 4 ways that it can transfer. What are the 4 ways? So it can transfer to the carotenoid, which is the another molecule that is there. So it goes up and then transfer to the carotenoid. What else? It can have a coupling between chlorophyll and carotenoid that can also dissipate the energy. Also, it can have the charge transfer between carotenoid and chlorophyll, or there will charge transfer between chlorophyll and chlorophyll. So there are 2 molecules, chlorophyll and carotenoids, and there are many permutation combinations that can happen. Broadly, we can divide it into 2 parts. One is energy transfer, and the other one is charge transfer. So charge transfer is where it is transferring the electron directly. In case of energy transfer, so it goes to the excited state and then comes back to the ground state. It emits the energy, and the next molecule absorbs that energy. So it's a dipole-dipole coupling. So this is the energy transfer. Now we will find out what is the process happening exactly in this system. Now let's go to the second point. What is protein organization in membrane? Now what we do is, this is the plant Arabidopsis thaliana. Now if we illuminate this plant with laser light, it will scatter a lot. And also there will be multiple signals. So to do spectroscopy, we need to take out the protein from these plants, from these leaves, and dissolve it into micelle, basically detergent, so that it covers like this and can be soluble in water. So to do this process, we need help from biotechnologists because we are spectroscopists. So nowadays, research is more interdisciplinary. So we collaborate with different people from different backgrounds, with biotechnology, with theoreticians, to understand some problems that is happening in nature. Now here we collaborate with University of Verona. So there they extract this protein, dissolve in the detergent. But as we know, protein has 3D structures. So depending on the surroundings it gets, it deforms. So the conformation will be different. Now to understand this in a better way, we took this membrane scaffold protein with lipids, and we put this protein which is CP29. I told you in the second slide that this protein is in between the periphery and the core. What it forms is, it forms a nanodisk. So we are trying to understand this protein inside this nanodisk, because this nanodisk mimics the thylakoid membrane I showed at the beginning, that discoidal thylakoid membrane with this embedded of photosystems. So this is more or less similar environment. So we want to have a protein organization which is very close to what it is inside the thylakoid membrane. The third point is carotenoid composition. We all know xanthophyll cycle, we studied in textbook. So basically where it what happens there, we have viola-xanthine, which is a carotenoid. It goes to the zeaxanthin when we have highlight. So what we did is expose this plant under highlight, so that through xanthophyll cycle it accommodates zeaxanthin. So we have zeaxanthin and we extract the protein and put it inside nanodisk. Now we have all these dots, right? Like protein organization in membrane, carotenoids, spectroscopic techniques. Now we want to connect all the dots. So we want to study these proteins through this spectroscopy. First is time resolved fluorescence. Now the red one is the normal protein in detergent. So it is decaying like this in like 12,000 picoseconds. So it is like 12 nanoseconds. Now when we go from detergent to nanodisk, it is becoming faster. So it is more coaxed. From there to zeaxanthin, so what is exposed in highlight, so that it is in photoprotection mode, it goes to the green. So it is more coaxed. So here you see, when we go from detergent to nanodisk, it is more coaxed, and then in GIA it is further coaxed. Now this chlorophyll is transferring somewhere in this structure. To find out where it is transferring, we'll be using transient absorption. So the pump probe one that I explained a little bit. So this is how the transient absorption spectra looks. Now in the x-axis, you can see this is change in absorption, delta A. And on the x-axis, we have wavelength. And we have spectra plotted at different time scale. So the first one, the pink, is 320 femtosecond. Then what happens? We goes to 10 picosecond, that is the blue one, to green to red, in nanosecond. So we change the time scale and we see how the spectra, the delta A spectra, is changing over time. Now, what are these signals saying? If we look at the chlorophyll B, so chlorophyll has two types of chlorophylls in the plant, chlorophyll A and chlorophyll B. Now chlorophyll B, as you can see, absorbs around 630, 640. So we pump this protein at 630 nanometers. So we are pumping selectively chlorophyll B. And at the beginning, we see the signal, the pink one at 320 femtosecond at 640. So this signal is very close to this, right? So this is coming from the chlorophyll B. So we have another signal at 680 nanometer. From where it is coming? If we look at chlorophyll A, you see the absorption here, 670, 680. This is coming from chlorophyll A. And if you look at this region, you see the red one, which is at the later time scale, not at the beginning. In nanosecond time scale, we see some signal coming up at 510. So this is carotenoid triplet. Now you can ask me why the carotenoid triplet is coming at later time scale. So now go back to the development diagram. I told you, so from singlet to triplet, the intersystem crossing is spin forbidden. So that's why it's a slow process. So we pump the chlorophyll, it goes to the singlet excited state, then it goes to the triplet chlorophyll, so it's a slow process. Then from triplet chlorophyll, triplet carotenoid is generating. So that's why it is taking time to generate. So we are observing this triplet carotenoid at nanosecond, not in femto and pico, because this is intersystem crossing and spin forbidden, it's slow. So from this one, what we can do is we can choose a particular wavelength. Let's see 680. So how this chlorophyll A, now this signal is negative. So this is bleaching. So basically This is ground state bleaching, how it recovers. If we plot the kinetics here, as you can see, the red to blue to green. So the recovery is becoming faster in nanodisk, which is very similar to what we have observed in fluorescence also. And also we can see in the 510 nanometer, where is carotenoid triplet, how the triplet is forming. You see this is growing. So in the red, where it is in detergent, the carotenoid triplet has formed more compared to when it is in nanodisk. And as you know, this triplet formation is not good for photosystems because it can generate the reactive oxygen species. So when the protein is inside the nanodisk, it is forming less reactive oxygen species. Now with this data, what we can do more? So this 2D data that we get from the transient absorption, if we feed this data with global analysis, what we get is basically we are fitting this data and we are getting decay associated spectra. So we get some signal at different time scales. And we try to analyze these signals at different time scales to know how the photophysics is happening. Now I will try to make it simple one. So in the DAS, if is ground state bleach or stimulated emission, if this is negative, that means it is decreasing. And if it is positive, that is mean it is increasing. So basically, decreasing to increasing means it is transferring from here to there. As we know, 630 is the chlorophyll B signal. So this is decreasing. And 680 is chlorophyll A signal, which is increasing. So what is happening is, if we excite it, then immediately at hundreds of femtosecond, it is transferring from chlorophyll B to chlorophyll A. So from here to there. Now in the next plot, the carotenoid signal here, this is because of the coupling between chlorophyll and carotenoid. Because we are pumping chlorophyll B at 630, we are not pumping carotenoid. But because of the coupling, we can see the carotenoid signal also. In the next spectra, as you can see, this guy is still going down. So still it is transferring in few picosecond also. But here you see, this is accepting, but we see a derivative shape here. What it means that this signal here, this one 680, it is going to the right. So it is shifting. That's why in the derivative shape is generated. So if the spectra is shifting towards the red, means the wavelength is increasing, so energy gap is decreasing. That means this is internal vibrational relaxation happening here. So we can see the vibrational relaxation in this time resolved spectra. In the next time scale, which is 84 picosecond, what is happening? Then the chlorophyll A is decreasing. Now it is in the negative. And what is happening? Now from here, we see there is a positive signal here. That means it is accepting this energy from this chlorophyll A to 510. That means it is carotenoid. Now which state of carotenoid? This is some state of carotenoid that we don't know. Now to find out this state, we did many measurements. I'm not going to the detail, otherwise you'll be bored. So we found out that this is carotenoid A star. So when we excite the chlorophyll, it is transferring the carotenoid star to dissipate the excess energy when there is more sunlight compared to the normal one. So in this state, it can dissipate the energy so that it does not destroy the organisms. Now you may ask that we are taking CP29 and it has many carotenoids. So namely 3. This is pioglitazanthin at L2, lutein at L1, and myosin at N1. Now we know the crystal structure of this protein. So we know the exact position of these carotenoids, and there are many chlorophylls around it. Now from the geometry, we know that myosin cannot transfer or take part in non-photochemical quenching. So it means it cannot take the energy from chlorophyll. So there are 2 carotenoids remaining. So this one, and the chlorophyll around. Now we should know which cluster is important in non-photochemical quenching. Then we can, the biotechnologist, if we can share this information to biotechnologists, they can tune the exact cluster so that they can improve the crop productivity. To understand which cluster is involved in this non-photochemical quenching, we again talk to the biotechnologist. What they did is they can do site-directed mutations. So they can remove a particular chlorophyll close to this carotenoid so that we disturb this cluster and see if the whole protein photophysics is disturbed or not. From there, we can know which cluster is important in non-photochemical quenching. So now, by this mutation, they removed a particular chlorophyll from this side and another chlorophyll from that side. So from close to this one and close to that one. Now, why we are interested in doing these measurements? Because when we looked at the literature, that means the published work in recent years, 2019, 20, with reputed journals, we found that it is not clear which cluster, whether A2 or A5, is important in non-photochemical quenching because there are contradictory reports. So by doing these mutations and with the help of these two spectroscopic techniques that I explained a little bit to you, that time resolved fluorescence and transient absorption, what we found out is that, to make long story short, that both the clusters are involved in this photochemical, non-photochemical quenching. So if we disturb A2 or A5, the protein is also perturbed. Means in non-photochemical quenching, this chlorophyll and carotenoid interactions are present in both the clusters. So if we can tune any of the clusters, then we can tune the photoprotection mechanism. Not only that, just to give you an idea, because in the experiments, we cannot see the individual chlorophylls. We can see the average signal coming from all the chlorophylls. If we can simulate the data we collected by the help of theoreticians, then we can exactly simulate which chlorophyll is more important in these processes. So exactly we can simulate our experimental data and get these processes that is happening between different chlorophylls and carotenoids. So just to give you an idea that by taking the help of biotechnologists, we get the protein, we get the mutations, we use the spectroscopy to find out the exact time scale, what is happening in femtosecond to picosecond to nanosecond. And again, by the help of theoreticians, we can exactly model which chlorophyll is transferring at what time scale. Now, I can skip this because it's already half an hour. So here you can see this is algae from ocean. It develops these blue iridescent colors at a particular life cycle stage. And why it is important? Because it helps to protect the photosynthetic apparatus. That I will not go into the detail. So far, in the last half an hour, what we got is we can understand the photosynthetic molecular mechanism, how the chlorophyll and carotenoids are interacting, and what are the exact chlorophylls and carotenoids involved in the process, so that we can tune the process to harvest more light when there is sun or even weather is cloudy, so that we can increase the crop productivity. Because if it harvests more, it can grow faster, and so that it can generate more crops. So I would like to thank all my collaborators and past group members for this, and thank you all for your attention. And if you have any question, please ask me. I'll be happy to answer. Thank you.
So please read the question. Please you can stop in the chat box or
I can't hear you properly. If you just write the question in chat, I will be able to answer.
now you can hear me now.
Yes, now a little bit better.
So I have one question. So this point where I think beta carotene is the linking state, right? I think you are that slide where you have shown chlorophyll A is more absorbing than other things.
Yes, I can hear you properly. I heard beta carotene. So yes, beta carotene is one of the carotenoids. So I just gave it an example. So like beta carotene, there are many types of carotenoids that is present. Let's say in plant, there are like violaxanthin, zeaxanthin. These are the carotenoids there. And you have different chlorophylls also, like chlorophyll A, chlorophyll B, and you know, nature changed the chlorophyll in a way, like in many organisms, there are chlorophyll D, chlorophyll A, chlorophyll F, which is more red shifted because they grow in caves and other parts. So if it is more towards the red, then the red light can penetrate more. As we know, wavelength is higher, so it can penetrate more. So it can harvest the red light more. So depending on the surroundings, this absorption spectra can be tuned or tuned by nature. So we are just trying to understand the nature.
Okay, yeah, that's what was my question. That let's say if it is in Italy or if it is in Indian little bit hotter region, when if there will be a change in this absorption spectrum or not.
Yes, but
You
this
answered answered that question that there will be a chlorophyll change, I think, or maybe plant changes its chlorophyll to take more red light
yes.
from the nature.
Yes. So depending on the environment, it will change, but it's a evolution. So it's a very slow process. It develops over many thousands of years, right?
Here you have taken only one type of plant extract, right? It's not done on so many plants or right?
Sorry, I can't hear you properly.
You have taken only one plant extract, right? Only one type of plant extract, right? So chlorophyll A or chlorophyll B extracted from the plant is only one type, right?
No, we are not extracting exactly one chlorophyll or one chlorophyll A or B. We are basically extracting a protein because in photosystem there are many light harvesting complexes like LHC2, CP29, 26. There are names like that. Now we took the CP29, the whole protein from there. Now inside this protein we have like 13 chlorophylls. Out of that 13 chlorophyll, we have like, if I remember correctly, 9 to 10 chlorophyll A and 3 chlorophyll B. So we chose a particular protein which we think is more important because it is bridging the peripheral proteins and the core proteins. So we focused in a particular protein and then studied how inside the protein these different chlorophyll and carotenoids are interacting. I mean, there are some other groups studying only chlorophyll extracting and studying the chlorophyll. But here our aim is not only to study the chlorophyll because this has been studied 30 years ago. Nowadays, we are trying to understand what is happening inside the proteins and when the protein is more close to the thylakoid membrane environment, not as extracted one in the detergent. So that's why we are making nanodisc, or even recently we are trying to do spectroscopy in even thylakoid membrane. That is a bit challenging because it is scattering a lot and also giving multiple signals. So we need to be analyzing many data sets. But the idea is to go to the natural point where the protein conformations are exactly what it is inside the plant.
Yes, I think it's clear now. Little bit of confusion is there still. So, okay, thank you, Chairman, for a very beautiful presentation. And I think we'll leave for lunch now.
Thank you. Bye.
Contact you later. Okay, bye. We will join again after half an hour.
হ্যালো, শোনা যাচ্ছে? হ্যালো, হ্যালো।
হ্যাঁ, হ্যালো, শোনা যাচ্ছে?
ওকে ওকে ওকে।
হ্যালো, জয়ন্তদা শুনতে পাচ্ছ তো?
হ্যাঁ 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 হ্যাঁ।
তাকে দুবার করে শোনা।
Hello. We are privileged to introduce our next speaker, Dr. Jayantu Nandu, a scholar whose contribution to supramolecular chemistry and biomaterials continue to inspire and advance the frontier of chemical sciences. Dr. Nandu currently serves as an assistant professor in Department of Chemistry at the University of North Bengal. His academic journey is marked by excellency and international engagement with prior appointment at Kalimpong College, IIST Shibpur as DST Inspired Faculty and postdoctoral fellowships at Netherlands and Israel. His doctoral research at Indian Association for the Cultivation of Science focused on synthetic amino acid and peptide-based gels, an area that laid the foundation for his current work in peptide-based cell replication system, supramolecular catalysis, and bio-inspired soft material. These themes not only probe the origins of life, but also open the new avenues in system chemistry and biomolecular engineering. Dr. Nandu's scholarly output includes more than 32 peer-reviewed publications in high-impact journals such as Nature Communication, Angochem, and Chemcom, with high index and his citation reflecting both the depth and influence in his field. He has successfully led multiple funded projects like DST Inspired, ACRB SRG, and UGC DAE CSR grant, and has mentored a growing group of PhD and MSc students. His affiliation with Indian Peptide Society and the Society for Polymer Science and the Royal Society of Chemical further underscore his commitment to collaborate and interdisciplinary science. His today's topic is self-assembly application on peptide synthesis. We look forward to his insight. Over to you, sir.
Screen is visible.
Yes, sir.
Is visible now? Hello?
Yes, it is visible.
Okay. Good afternoon, everybody. First, I want to thank Dr. Shukdev Ghosh, Principal Sir of Sri Krishna College, and HOD, as well as all the faculty members of Department of Chemistry for arranging this beautiful seminar and inviting me. I want to thank the joint conveners, Dr. Paramita Hajra and Dr. Nabodhuti Bodman for the invitation and arranging the seminar to inspire and motivate the college student. Okay, so today I will talk about amino acid and short peptide based self-assembled material. So first I will talk about self-assembly. Self-assembly is a spontaneous association of molecule or building block and the structurally well-defined aggregate. This is okay, now it's okay. Okay, spontaneous association of molecule or building block into stable and structurally well-defined aggregate under the equilibrium condition. Biology often use hierarchical self-assembly process to form complex functional structure from smaller component. At each level, there are different kind of non-covalent interaction present and which brings the subunit of lower order molecule and to form the complex molecule. What are the non-covalent interaction present at here? What are the is visible now? Okay. What are the What are the non-covalent interaction present at here? Like hydrogen bonding interaction, aromatic interaction, ionic interaction, hydrophobic interaction, underall interaction. These are the different kind of non-covalent interaction. I have shown here some of the features. Like here the hydrogen bonding interaction in our class and in our class 10 or 11 class, that hydrogen bonding interaction among the water molecule. These are the aromatic interaction between the pi conjugated molecules. These are the ionic interaction between the positive charge and the negative charge. Okay. So if you see the molecules are connected by the atom. They are the covalent bond. Okay. And this molecule undergoes supramolecular assemblies, and this has been done using this kind of non-covalent interaction. And when they form these supramolecular assemblies, there are the emergence of new functional properties, which was not present in the individual molecule. But when they are aggregated using this non-covalent interaction, they form kind of supramolecular assembly and they exhibit different new functional property which was not present in the individual molecule. You can see here, they are the different Legos. Okay, they are the different Legos, and using these Legos, children, house, children makes building. Okay, they are happy when somebody give them, so they are trying to make this kind of building block. And in nature, there are also several building blocks. Like if you see, the nature use protein, carbohydrate, nucleic acid, lipid, and there are building block at the like amino acids, sugar unit, and this is the nucleotide. Okay, fatty acids. These are the nucleotide, and nature use this building block with having some particular chirality, and using that particular molecule, they construct different large biopolymer like protein, carbohydrate, nucleic acid, lipid, and in some cases that hetero biopolymer like composed of sugar and nucleic acid, sugar, amino acid, and sugar and nucleic acid, and different kind of mixed kind of biopolymer are formed. So nature use this building block to make the functional biomolecule. In my talk, I will talk about amino acid. I will talk about amino acid. Okay. So if you see, this is the structure of the alpha amino acid. Why it is called alpha amino acid? If you have amino acid, it has two ends. One end is carboxylic acid and another end is amine. Okay. So carboxylic acid has higher priority than the amine, as this carboxylic acid is attached to this carbon. So this carbon is called the alpha carbon. This carbon is called the alpha carbon, and this is called the alpha amino acid. Okay, and it has R group, and depending upon the R group, their functional property, their self-assembling property are varied. Okay, I have drawn other two structure at here in the right side. So this structure is called beta alanine, and this structure is called the gamma amino butyric acid. Okay. So in this structure, if you see, the amine group is connected in the beta position. So this position is alpha and this position is beta. So this is a beta amino acid, and this is called the beta alanine. Okay. It has similar molecular formula with alanine. So it is called the beta alanine. In the second, this amino acid is called GABA. GABA means gamma amino butyric acid. How many carbon is present at here? 1, 2, 3, 4. 4 carbon is present. 4 carbon means it is but. So 4 amino or gamma amino butyric acid. This act as a neurotransmitter in our body. Okay, so I have shown here the natural alpha amino acid. You can see here they are marked with a different color. Specifically, they are hydrophobic. Some are aromatic, like the phenylalanine, tryptophan, tyrosine. They are aromatic. Tyrosine, they are aromatic. Some amino acid have acidic, like aspartic acid, glutamic acid. They have acidic side chain. Some amino acid like lysine, arginine, histidine, they have a basic nitrogen part, and some amino acids are also neutral. Some amino acids are aliphatic. So these are different alpha amino acid, and nature use this amino acid to construct different functional biopolymer molecule. So how peptides are synthesized? So I have drawn here 2 amino acid, okay, 2 alpha amino acid. So I mark at A, and this amino acid mark as B. So how to form peptide bond? So there will be a condensation reaction and the loss of water, and it will form a peptide. It will form a peptide bond, CONE. This peptide bond will be formed. Okay. So if I synthesize AB peptide, if I want to synthesize AB peptide, so how we can synthesize this? So here there will be a condensation and we'll get AB. But the system is not simple like that, because these amino acid A, they can also condense to form AA, this amino acid B, they can also condense to form BB, and another sequence, the reverse sequence of AB, that is BA, can be formed. So we need to use different peptide coupling agent and different methodology like solution phase methodology and the solution phase methodology. Okay. So if you want to synthesize some ester, if you have a carboxylic acid and if you have a alcohol, what we can prepare the ester? We took these two compound, and in presence of acidic environment, if you reflux it, we get ester. So similar analogy, there is NH2 instead of OH, so that can form the ester bond, but actually this will not form the ester bond. What will happen? If you see, this is an acid and this is an amine base, so they will form a salt. They will form a salt when you mix them and heat it. They will form a salt. So they will transform into the carboxylate, and this will be the NH3 plus, that means ammonium ion. So when they form carboxylate and the ammonium, so they are making salt. From that, you will not get the peptide. So what we have to do? We have to make the OH group as a good living group. So for that purpose, we have to use some peptide coupling agent, which will peptide coupling agent or other activating agent, which will convert the OH group to a good living group. Then the NH2 will attack at here and we will get this peptide. Moreover, we need protecting group. Otherwise, there will be a random reaction. So to couple between these two groups, we need to protect the N-terminal and we need to protect the C-terminal. We need to protect the C-terminal. Then you have to use the peptide coupling agent. Then we'll get this compound protected dipeptide. And depending upon our requirement, we can remove the protecting group. What is protecting group? Protecting group are the group which can be easily removed, which can be used to protect some functional group during the reaction, and after the reaction, this group can be easily removed. Like in case of the NH2 group, we use the Bock group, EBMOC group, and for the carboxylic acid group, we use the methyl ester, ethyl ester, benzyl group, some other group. So in this method, we can synthesize peptide. Okay. So there are the two methodology. One is called the solution phase methodology, another is the solid phase methodology. So you will learn in your BSc class during the course of the time what are the solution phase methodology, what is the solid phase methodology. So I am not going there. So now question is, why we will study the peptide based material? So I give you here 3 points. Number 1, to understand the different living system and the way of function. So in our body, there are lot of molecules which are peptide based. There are the large peptide, large protein, enzyme, hormone. Okay. So these molecules are based on the peptide. So these molecules are synthesized in our body. So we take protein. We take when you take meat, when you take egg, when you take pulse. So that contains protein. And in our body, these molecules are digested and converted to the different amino acid. And our body, depending upon the requirement, they synthesize the molecule. They synthesize different enzyme, different hormone, different large protein for a particular purpose. And when this purpose was done, so there are some other enzyme also working which chop up this molecule and the molecule are excreted from our body. So if you want to learn some living species, how they are functioning, how they are working, so you have to know how the protein synthesis in our body, how they are functioning, the cell activity and others. Another point is to study the different neurodegenerative disease. Like when we see in our societies, some old man forget his name, some old man forget who is sitting nearby him. Maybe he is his son or her daughter or her wife, sorry, his wife. So he cannot remember. So this disease is called the Alzheimer disease. And we see somebody cannot walk properly, cannot write properly. So these diseases are called the Parkinson disease. So there are a lot of disease which is caused by the different protein. So we have to learn how these protein are misfolded and form this toxic material which is affecting our nerve cell. So nerve cell and central nervous system, our brain, that's why we forget all these things. So first point is to know the living system, how they are working. And second point is how this neurodegenerative disease is affecting the human and their activity. And third point is peptide-based soft material for mankind. So you can synthesize different kind of synthetic short peptide, and you can use this peptide for the benefit of the mankind. It can be used for the drug delivery, it can be used for the tissue engineering, it can be used for the wound healing, it can be used for the 3D fabrication, it can be used for the bone repairing. So if you design in a proper way, so this peptide can be used for the beneficial of the mankind. Okay, so we are talking about in general application of the peptide. So I will now talk about a little bit around my research. I mainly work on the short peptide based self-assembly, and we synthesize different short peptide amino acid derivative and we study their self-assembly. So if you take some compound, some peptide and dissolve in some solvent, in solvent, it can be water, it can be phosphate buffer, it can be organic solvent, and if you heat it, it will be dissolved. And when we cool the solution, what will happen? These molecules are coming closer together. So most of the time, they will form precipitate. But in some of the cases, they will form crystal. And when there is a good hydrophobic and hydrophilic balance of this molecule in that particular solvent you are studying, they can form the gel molecule. So if you see how the gelation form, suppose these are the gelation occur, suppose these are the molecule. So if you use some trigger, this trigger may be the alteration of the pH, this trigger can be the use of sonication, this trigger can be use of the salt or some other tricks. So what will happen? These molecules are coming closer together and forming the fiber, and they are forming lot of fiber, and within that fiber matrix, the solvent what you are using at here, that will be trapped, and when it will be trapped, it will restrict the flow of the solvent. As a result, you can invert it. Okay, so if you take water in a glass vial and invert it, so water will fall at here. But when it is forming gel, so the solvent, the flow of the solvent will be restricted and it will form a gel. So what type of gel you are preparing? There are the different classification. If you prepare a gel which is based off the natural material, so this is called natural, like aloe vera gel. Aloe vera gel is a natural gel. Otherwise, if you synthesize by artificially, like using that molecule which is present in the nature, so this is artificial gel. In which medium it is preparing? If it is organic solvent, so this is called the organogel. If it is prepared in the water, aqueous solvent, so this is called the hydrogel. And another thing is called the aerogel or aerogel. If you remove the solvent matrix, so what will happen? there will be the matrix will be there. Okay, if you remove the water, so what will be present there? There will be matrix, and this position will be filled by air. So sometimes it is called the aerogel. Okay, so in the artificial gel, what was prepared in the laboratory, that are two kind. One kind is supramolecular gel. Supramolecular gel, basically the physical gel, which is prepared using the different kind of non-covalent interaction. Like if you change the pH, the gel will be break, and if you again back to the pH, so gel will be reformed. And most common is the heating. If you heat the molecule assembly will be break and converted again in the monomer, and again when you are cooling, so it is forming gel. So these are the physical stimuli gel. And there are the another kind of gel that is called the macromolecule, which is mainly done by polymerization reaction using the different polymerizing agent. So in the in this presentation, I will talk about single amino acid based self-assembling systems. And in the next part, I will talk about design and development of artificial helical system and their helicity tuning. Okay. So the motivation in this research, that there has been previous studies that which suggests that peptide-based fibril can be formed using the different kind of non-covalent interaction like hydrophobic interaction, hydrogen bonding interaction, pi-pi stacking interaction. And it has been observed by several groups, and the pioneer work was done by Ehud Gazit from Tel Aviv University, Israel. He postulated that the possible role of pi-pi stacking interaction in the self-assembly of amyloid fibril formation. Suppose this is a peptide sequence and which contain an aromatic amino acid, and this aromatic amino acid are stacking through pi-pi stacking interaction, and they are forming the fiber. Okay, so after that, there are people are studying different neurodegenerative peptide. Okay, like I talk about the Alzheimer disease. Alzheimer disease is caused by this peptide sequence called the A beta amyloid, and it contains 42 residue. So people are trying which part is responsible for this disease, like through the top-down approach, which part is responsible for this amyloid fibril formation. So initially people thought about the whole peptide is responsible for the toxic fibril formation. After study of the different peptide sequence, they found that this KLVFF, this is they call the central core of the peptide, and this is responsible for the fibril formation. And it has been observed that this peptide sequence also forming the fibril, which is toxic towards our neuronal cell. So they studied KLVFF, and again they are studying the sequence, and they found that FF means phenylalanine. The phenylalanine one letter name is F. This dipeptide is toxic. So they studied, this group, Gazit group, they studied that this dipeptide can form nanotube structure. Okay, this is acyclic, but they can form the cyclic nanotube structure, and they have studied by TEM, transmission electron microscopic study, and they found that this dipeptide can form nanotube. Okay, and after study, they have also found that single phenylalanine, they are also responsible for formation of toxic nanofiber like the large peptide sequence. Okay, so if we change if we change the Okay, if you change the aromatic interaction at here, we can modulate the self assembly, and in this regard we have wrote a review on phenylalanine based fibrillar system. So when we take our food which containing phenylalanine, in our body there is one enzyme, it is called phenylalanine hydroxylase. What is the role of this phenylalanine hydroxylase? Phenylalanine converted using the phenylalanine hydroxylase to tyrosine, that is 4-hydroxylation take place, and this tyrosine later further converted to the dopa, it converted to the dopamine. So this process is very important. What happened in the pregnant woman, that in some of the cases they have high phenylalanine content, and their body, due to some gene mutation, phenylalanine hydroxylase was not working. So what will happen in their body? The concentration of phenylalanine will be increased, and they will not convert it to the following pathway like tyrosine, dopamine, and other. So as the phenylalanine concentration will be increased, so this will be transferred to the baby, and as a result, what will happen, that baby have some mental retardation. So this disease is called the phenylketonuria. This disease is called the phenylketonuria. So we have studied, we have written a review, and we discuss how phenylalanine in presence of different stimuli like pH, ionic strength, solvent concentration, they can form the toxic fiber, and by which means, like in presence of nanoparticle, in presence of drug, in presence of polyphenolic compound. Polyphenolic compound is very important to stop the amyloid-like fibril formation. Okay, Alzheimer's disease formation, in some of the paper they have reported polyphenolic compound, in presence of polyphenolic compound, this fibril formation is stopped, and they form some non-toxic small molecule aggregate. Okay, so we discuss how phenylalanine assisted in a fibril formation, and by help of different agent, how this phenylalanine based fibril formation are stopped. So next, we think about if we modulate the ring incorporating different functional group, it can affect the self-assembly process. In this regard, it has been observed that if we incorporate some halogen molecule at here, so they can affect the self-assembly process. Okay, so it has been observed that this molecule have 2 aromatic residue, and when they incorporated bromine, chlorine, iodine, they observed that the halogenated derivative has a better self-assembly, and they are forming gel. Similarly, this is the EBMOC phenylalanine, and another group by Nilsson, they have incorporated halogen group, and they have noticed that if you incorporate more number of halogen and different position, it can affect the self-assembly process. And in this direction, we thought about if we do incorporate some functional group like nitro in this ring, it can affect the self-assembly process. And if we incorporate it in the peptide sequence, this nitro group can influence the self-assembly. So we try to synthesize 4-nitrophenylalanine. So we have taken phenylalanine, and we have done nitration in the using the concentrated nitric acid and concentrated sulfuric acid. We got this 4-nitrophenylalanine. So this compound formed crystal in water, including a water molecule, and we want to try to do some reaction in DMSO medium, and we found that when you dissolve this compound in DMSO, it is forming the reddish brown color gel. So we think about DMSO and water, both are soluble. How their self-assembly affected if you change the solvent? Because in DMSO, they are forming gel, and in water, they are forming crystal. So we vary the ratio. When we vary the ratio, we find that in each case we are changing the 10%, and we have seen that 100%, 90%, and 80%, in these three cases they are forming self-supported gel, but in other cases they are forming crystal. We think about why this color is generating. We have studied the UV visible spectroscopy. So when we do the UV visible spectroscopy, we found that a peak around 480 was observed, and this peak is due to the charge transfer complex formation between electron deficient 4-nitrophenylalanine and electron rich DMSO. And when we increase the percentage of water, what we observe, this peak is diminished, and this the main peak was also blue shifted. So here we have concluded that depending upon the solvent composition, in one case it is forming the gel molecule, in another case it is forming the hydrogen bonding mediated crystal. So depending upon the composition, final self-assembly is dependent. Later, we have added aromatic amino acid in this matrix, and we find that the color is more intense. That actually indicates that when we incorporate this aromatic amino acid in this matrix, they are also take part in the charge transfer interaction. As a result, the color became intense. So we have also modified tyrosine molecule, and we have prepared 3-nitrotyrosine. And sometimes when people ask why the color of urine is yellow, we commonly answer that it contains bilirubin and biliverdin. And the third person which is responsible for this color is 3-nitrotyrosine. So 3-nitrotyrosine is formed in our body in presence of reactive nitrogenating species. So 3-nitrotyrosine is formed in our body. So what we have done, we have attached some EBMO group. EBMO group is an aromatic group which assists in the self-assembly. What we observe that this EBMO conjugated 3-nitrotyrosine form gel in a wide range of pH. And if you see, when you change the pH, the color is changing. And we find that this molecule, if you see only this molecule, this is a orthonitrophenol. This is orthonitrophenol, and the pK is around 7 or 7.2. So below this, the color is light, and above this, the nitrophenol is deprotonated. So there is a more conjugation, and we get the more intense color. And this material has been used for as an antimicrobial agent. So we have also used tyrosine. Tyrosine, it forms gel in DMSO water, and we have used this matrix for the charge transfer interaction with DDQ. DDQ is a highly electron deficient molecule. When it was incorporated in the matrix, it will restrict the pi-pi interaction among the tyrosine molecule. As a result, a new reddish color is formed. You can see at here, this is the tyrosine gel, and this is the tyrosine and DDQ in the 1:1 ratio. This tyrosine self-assemble to form this fiber. But when they form the charge transfer complex, due to the interaction of the tyrosine and the DDQ, the self-assembly is broken, and they form this kind of debris structure. And we have developed the EBMO phenylalanine based antimicrobial agent. So this is the normal phenylalanine. When it was attached with the EBMO group, it is formed the hydrogel. In a paper in 2015, one author comment that the flexibility part of the aromatic amino acid is very important. So to address this, we have changed the flexibility of the amino acid. What we have done, we remove the aromatic group and we make it a cyclohexyl alanine. And in the second molecule, we remove the flexible CH2 unit and we remove we increase the flexibility. So this is CHA, this is the PHG, and this is the homophenylalanine. And we have studied the self-assembly in the similar condition pH 7. And we find that all these three compounds are forming gel. As well as that, we check their antimicrobial activity, and we find that compared to the natural molecule, these three molecules are better in antimicrobial activity. But when we check the homolytic cleavage activity in presence of globin, so we observe that the PHG molecule is better than this. Okay, so these are the conclusion. So I will talk about in the next part helical system. So we observe helical architecture in our body, in our nature everywhere. Like you can see the neutrino molecule, DNA double helix. We can see this from the plant, the galaxy, and this is the tobacco mosaic virus. This is the flower, snail. This is the seed cell. Okay, we observe different helical systems. So inspired by this kind of architecture, we have in the nature, we have DNA double helix, RNA helix, collagen triple helix. So we synthesize one molecule dipeptide. This is composed of naphthalimide, and it is an achiral amino acid, and this is a chiral amino acid. The plan was to use some group instead of EBMO group. EBMO group is commonly used. So instead of EBMO group, I want to use this naphthalimide group. Moreover, naphthalimide is a highly fluorescent molecule, and the peptide molecule you can synthesize easily, and you can modify the system easily. So when we study this molecule, that is N is equal to 2, that is beta alanine, and this is the isoleucine. This self-assemble in the DMSO water forming the fluorescent gel. When we check the morphology, we find this is the left-handed helical nanofiber. We thought about the modulation of supramolecular helicity. In our nature, sometimes the DNA, they also there is also tuning of the helicity in the disease state. So we synthesize in the first system, we have kept N is equal to 2, and we have changed the chirality of the system. This is the very common practice. So what we have done, we changed the chirality of the valine. This is the valine amino acid. Initially we have used L, then D, and we introduce another molecule which is very similar, but this is achiral. This amino acid is called the 2-amino isobutyric acid, is abbreviated as AIB. This is achiral, so I have put it A. So this compound form gel. The chiral molecule, they are forming gel, but the achiral molecule, they are not forming the gel. So we studied their self-assembly using the UV visible spectroscopy, and what we have done, we have taken 40 micromolar compound, and when we increase, this gel was studied in the DMSO water medium, using the DMSO water medium. So what we have used, so initially we have studied in the DMSO, and when you increase the percentage of water, we find there is a red sheet in every cases. So this is called the J type of the aggregation. In the J type of aggregation, the chromophore, there is a slippage, and we have studied their fluorescence. We observed that initially there is fluorescence very weak, but when you increase the percentage of water, we saw there is a jump in the fluorescence intensity. This is called the aggregation induced enhancement in the emission. So interesting thing is the circular dichroism CD study. For NLV and NDB, they have a mirror image, but the NAA, which is passing through a zero line, you can see here some dotted, some light color is there. So this molecule contains a chiral molecule, but when it was dissolved in a DMSO, they are not forming any aggregate, they are in the monomeric state. But when you put water, they are forming gel, they are forming a supramolecular aggregate, and this supramolecular aggregate was characterized or recognized by the CD spectroscopy. So you can see here, for the L, this is the blue line. For the D, This is the red line, and for the achiral molecule, this is passing through the middle. So we check the morphology. For the NLB, we got the left-handed helix, and for the NDB, we are getting the right-handed helix, and for NA, we are getting the flake-like morphology. So this is the table of content. You can imagine that this molecule has a chiral molecule, and this is a knob of a radio. So when you change the radio station, we are able to listen different program in that station. So when you change to L, when you change to D valine, and when you change to the achiral amino acid, we are getting the different kind of morphology. So in the next system, what we have done, we keep the chiral system fixed, and we increase the, what we have done, we have increased the achiral amino acid. So when n is equal to 1, this is glycine. When n is equal to 2, this is beta-alanine, then GABA, and so on. So what we observe, that N1, that is glycine containing molecule, is not forming gel. It is a solution, and after that it was precipitating. But N2, N3, N4, N5, they are forming gel. Okay, so you can see from 90% to 50% at 3 millimolar concentration, they are forming gel. So we have studied their CD spectroscopy. What we observe, that here N2 means there are the two number of methylene unit. Okay, N2, N4 have an even number of methylene unit, and N3 and N5 have an odd number of methylene unit. So this having the same chiral center, but when you change the number of methylene unit, their CD pattern is changing. Like N2 and N4, this blue color spectra, this spectra is in the negative direction, first peak in the negative direction, and this red and the another color, they are in the opposite direction, that is the positive direction. That indicates that even you change the methylene unit, their supramolecular helicity change, still they have the same chiral center, same L isoleucine. We have checked their morphology. For N2, we are getting the left-handed helical nanofiber. For N3, we are getting the right-handed helical nanofiber. So we have studied their morphology and compared the helicity with the CD. So N2 negative left-handed, N3 positive cotton effect right-handed. So even you change the methylene unit, N is equal to even. So we are getting the left-handed, N is equal to odd, we are getting the right-handed image. So we can modulate the supramolecular helicity not by changing chirality, by changing the achiral part. And this is the another interesting work. In this work, we have changes the supramolecular chirality by changing the solvent composition of achiral solvent, not chiral, achiral solvent. So here we have studied the time-dependent behavior of N2 molecule. So here N is equal to 2, that is beta-alanine. So when we study in the longer duration, okay, so initially it is forming gel within 2 minutes. When we go for 1 hour, 2 hour, we observe that these gel are broken and they are forming crystalline precipitate. The CP represents the crystalline precipitate. What is happening? So this is the DMSO solution. We are putting water, and this is the forming gel. So with time, they are forming the gel crystal. Within the matrix, you can see some crystal is forming. And after 1 hour, so they are precipitating as crystalline precipitate. So we studied here at the 90% water composition. So we thought about if we decrease the water percentage, how they are shelf assembly, how they affect. Okay, so we study the CD spectroscopy, and we study in the 45%. So initially in the 45% water DMSO, rest is DMSO, we saw some line passing to the 0, but with time this peak is growing. So this, the first peak at here is the right in the top positive direction, Cotton effect in the positive. But if you see this spectra, we have also studied N2 molecule. This N2 molecule direction is negative direction. So that indicate that with the change of solvent composition of DMSO water, there is a helicity tuning. So we check the full range, we observe that up to 55%, the peak is the positive direction, and after that, the peak is in the negative direction. So we thought whether this is the effect of chirality, this is the effect of solvent. So we synthesize the D-amino acid based molecule, and we observe that in case of the 50% water, the peak is in the negative direction. But when you go to the 60%, the peak is again changed to the positive direction. So that indicate this helicity tuning was due to the solvent composition. So we studied the self-assembly at the 40%. We observed that they are forming the small particle and they are forming the right-handed helical nanofiber. But in case of the 90% water, or this is the same image and same image, we are getting the left-handed helical nanofiber. So you can say that depending upon the solvent, when you have a more water, it giving the left-handed helical nanofiber. When you have a less amount of water, it giving the right-handed helical nanofiber. So this work has been accepted in small and recently accepted as a front piece in the small. So in the last work, what we have done, we have incorporated some electron donating group at here, and we have started the self-assembly in the DMSO water 90% and chloroform water, chloroform MCH. Okay, so the compound is dissolved in the DMSO, we put water, and the sample is dissolved in the chloroform and put MCH. Okay, so we observe that when we study in this condition, we observe that they are forming gel. We studied their morphology. Here we are getting the mixture of both left and right-handed helical nanofiber in the both cases. This has been studied in the DMSO water. We are getting both left-handed and right-handed, and we are studying the MCH chloroform condition, and we are getting both left and right-handed. This compound is highly fluorescent. We have checked their cell viability, that this is good, and we have used this molecule for the cell imaging. And this is the TOC. So we dissolve the molecule in a suitable solvent, and when you put more polar solvent, water, so in this case we are getting the J type of aggregation, and in this case we are dissolving in the chloroform, and we are putting extremely non-polar MCH, and there is a gel formation, and the self-assembly type is J type of aggregation. So depending upon the path, depending upon the solvent, we are getting from same molecule, we are getting the J type of aggregation and H type of aggregation. So we have discussed various amino acid based self-assembling system. We have discussed NMI based conjugated system, their self-assembly, how we can tune their helicity. So I want to thank my student, Soubhik. Piyush Soubhik already got the degree. Piyush recently submitted. There are my present student. I want to thank DST Inspire, ACRB, SRG, and UGC DAE for financial assistance. I also thank all the collaborators. Without their help, I cannot do work. Thank you, everybody. Thank you for your attention.
I think you had that clap from our side. Hello? Am I
Hello.
audible?
Yeah, audible.
Yeah, you had that clap, right? So you presented very nicely. I think our students understood everything. So let me take some questions and then
Yeah. Can you stop the presentation?
Yes. Yeah, you can stop the presentation.
Okay. If you turn on the video, it will be better. Okay.
I think that's
Hi Monglu, how are you?
Hello, am I audible?
Yes.
Okay.
I cannot hear you.